Welcome to this episode of the Dry Eye and Ocular Surface Disease section of the Clinical Podcast Series, brought to you by the American Academy of Optometry Foundation. Today's episode is Culture and Non-Culture based approaches reveal unique features of the ocular microbiome in dry eye patients. My name is Kaleb Abbott. I'm an optometrist and an assistant professor of ophthalmology at the University of Colorado School of Medicine. I practice in our dry eye clinic, and I'm joined by Dr. Mila Ioussifova, who is the owner of South Waterfront Eye Care in Portland, Oregon. She's a certified nutrition specialist, fellow of the Ocular Wellness and Nutrition Society, and an expert in dry eye and the gut and ocular microbiome, and a total thought leader in the field of dry eye. I'm thrilled to be able to pick her brain today on this fascinating topic. Thank you for being here.
Thank you for having me.
So you are an expert in the field of microbiome. You know a lot about the gut microbiome as well as the ocular microbiome. Can you give us a little background on what the ocular microbiome is, especially as it pertains to dry eye and ocular surface disease?
So we have to think of the ocular surface just like any other mucous membranes in terms of the immunological function. And how similar it is to the gastrointestinal environment. Just like we have our gut microbiome, microbiota in the gut, we have a microbiota on the ocular surface. And now we have several studies have shown that the innate immune system of the ocular surface epithelium can differentiate between pathogenic and non-pathogenic bacteria. So the commensal, like the beneficial bacteria, contribute to maintain the ocular health by inhibiting apoptosis, barrier preservation, just like they do in the gut, and by interacting with the host immune system.
Great. So what was the purpose of this study in particular?
So the purpose of this study was to characterize the ocular microbiome in the individuals with either no self-reported symptoms of dry eye or with varying degrees of dry eye disease symptoms. And they wanted to identify the compositional changes in the microbiome that were characterized by dry eye disease.
So how was the study actually performed?
So they collected samples from the lower conjunctival sac of tear samples of 91 individuals, and 61 of them had dry eye and 30 of them were healthy. And the samples were used in both culture-dependent analysis and next-generation sequence and non-culture. And participants were characterized as normal, which again were 30. These were students from and staff from the university. And then they recruited the patients with dry eye from the Norwegian Dry Eye Clinic.
Okay, and what did the study end up finding?
So this week I can really geek out on, so I'll try to kind of keep it as condensed as possible. Yes. So there were a total of 190 colonies and 6 major genera were identified. And they found the most abundant genera to be Staphylococcus, Corynebacterium, and Cutinibacterium. So Corynebacterium, depending on the species, was found to be either positively or negatively associated with dry eye disease, which actually kind of similar in the gut too. There are a lot of bacteria, depending on the species, could either be protective or hurtful, or they could be pathogenic. And then they also found the Blottia genus to be the most associated with dry eye disease. So given the high abundance of Corynebacterium detected on the ocular surface in the study and previous work, future studies could investigate how this genus, this particular one, can interact with the immune system and its potential effect on ocular health. So while this study suggests, an association and not causation of dry eye, it's just interesting to see these two particular Blottia and Corynebacterium to be associated. And these are kind of, I think, good candidates to be investigated further in the future in the role of ocular surface disease and how we can modulate the composition or the dysbiosis, the imbalance of the microbes on the ocular surface when we're treating dry eye disease.
It's totally fascinating to think that the dry eye patients that we see every day generally have a different ocular microbiome than our healthy patients. And this is something that we're obviously still learning quite a bit about, and we're trying to figure out how is this going to, in the future, play a role in our treatments in patients like this. So this is all quite nuanced, especially for those of us that are not researchers and we're just in clinic all day. What do you think the relevant take-home points are for us clinicians?
So, as clinicians, it's important for us to, first of all, recognize that there is an ocular surface microbiota. And just like now we have so much evidence on the role of the gut microbiome in pretty much every non-communicable disease. So when we're treating now diabetes and heart disease, especially autoimmune conditions, we know that there's gut health that has to be addressed. So similar analogy, similar thought has to be on the ocular surface. So we have to be mindful of what's going on with the ocular surface when we are prescribing certain therapeutics, topical antibiotics especially, or if we're using contact lenses. So a lot of changes and disturbances could be happening on the ocular surface microbiome. And again, this is going to sort of pave, so more studies are needed, but I think we're going to have that tailored approach to modulating the not only gut microbiome when we're dealing with dry eye patients, but also the ocular surface microbiome.
Well, thank you so much for really delving into this topic today. So special thanks to Dr. Mila Ioussifova for joining us today on the Clinical Podcast Series, and we'll see you at the next episode.